Hello everyone and welcome to our latest MDPI webinar on molecules entitled The Chemical Biology Research in France. This is the third webinar on molecules based on special issues from our journal of the same name. If you are interested in submitting your paper to the special issues that are linked to this webinar, please check the webinar's webpage. I would like to remind everyone that this webinar is recorded. The recording will be made available as soon as possible and will be posted on the webinar's webpage. The webinar is also being live streamed on MDPI's YouTube channel. Hello to those of you watching us on there. Furthermore, certificates of attendance are available upon request to molecules.webinar@mdpi.com. We can only prepare certificates for those of you registered and watching on Zoom, not on YouTube. Today's webinar is chaired by Dr. Paola Arimondo, Director of Research at the Institut Pasteur-CNRS. She studied chemistry at the University of Pisa and received her PhD in biophysics at the MNNHN in Paris. Her research focuses on the interactions between nucleic acids and proteins and their modulation by small chemical molecules. Initially, she developed a strategy to target a specific DNA site in a class of anti-tumor agents directed against human DNA, topoisomerases 1 and 2, resulting in site-specific DNA scissors. After a 6-month sabbatical in 2005 at the University of California in Berkeley, she initiated a new project developing an innovative chemical strategy aiming at the epigenetic control of gene expression in cancers. The chemical modulators of the epigenetic regulation are also used as probes to better understand the role of DNA and histone methylation in cancer and other human diseases, and identify new targets or biomarkers. Paola Arimondo is also co-author of 123 publications and 10 patents. Thank you very much, Paola, for accepting to chair this webinar. There will be also 3 presentations today given by Dr. Paola Arimondo, Dr. Frédéric Taran, and Dr. Andrey Klymchenko. The presentations will be followed by a Q&A moderated by the chair. If you have any questions for our speakers, please feel free to post them at any time in the Q&A box, which you can find at the bottom of your screen. Please make sure to add the name of the person to whom you're addressing the question. The time that I hand over the microphone to Paola, and hope you all enjoy the webinar.
Thank you very much for the kind introduction. So thank you very much to Molecules to have organized this webinar that is dedicated to the chemical biology research in France. As Cédric said, there's a special issue dedicated to chemical biology research in France that will be published as an open access journal in Molecules. So if there's slots available, so don't hesitate to contact us. Why during this type of special issue? Because the chemistry at the interface of biology has always played an important role in the research in France since the 19th century, with the discoveries made by Louis Pasteur and the application to medicine and biotechnology. So chemical biology opens many possibilities of developing new cutting-edge technology, as we know, to observe and understand biological questions and discover, for example, new treatment or and other solutions. So I'm just trying to see how to move forward, sorry. So I will start by presenting what we do in the laboratory. Then Frédéric Taran will present bioorthogonal reaction with mesoionics. And then Andrey Klymchenko will then show the setup bright fluorescent light-up probes for biosensing and bioimaging. And then there will be a discussion at the end. So I will start by presenting, so my laboratory works on the design and use of chemical tools that target the epigenetics, and the aim is to reprogram the cells which have an aberrant epigenetic modifications. Why we develop chemical tools. So there was a great breakthrough in the years 2000 with the discovery of bioorthogonal chemistry and the development of chemical reaction that occur in living systems. And Frédéric, for example, will show you new bioorthogonal reaction with mesoionics. The other advantage of using chemical tools, thanks to this bioorthogonal chemistry, we can now use these chemical tools to observe and study all types of biomolecules, protein, lipids, nucleic acids, glycans, in living systems, in cells, tissue, and animals. And Andrey will show you how he used bright fluorescent light-up probes for biosensing and bioimaging. The advantage of using chemical tools is that they can reveal a function of a specific molecule without suppressing the entire molecule. It's what happens when we use siRNA, CRISPR, or without overexpressing it. This is what we do when we use genetic tools. So this is a great advantage. We don't alter the physiological situation. These molecules are also reversible. They're rapid, tunable. We can wash them off. And so it gives better spatial and temporal control. In addition, they don't necessarily disrupt the entire interactome of a biomolecule in the biological context. When you suppress a protein in a complex, you can disrupt the complex. If you inhibit an interface, for example, protein-protein interface, maybe you can keep the complex intact and study the protein complexes. What's becoming more and more interesting for understanding the biological mechanism. Of course, there's also some disadvantage of using chemical tools. We definitely need to have compounds that are single target specificity to have surgical tools and decrease the side effects. We need to have high quality and cell active compounds, and that's not easy. And we need maybe also to make them context specific. I mean, direct them to specific genomic sequences. And maybe we can combine these with the DCAS systems. So in my lab, as I was telling you at the Institut Pasteur, we develop chemical tools that target the epigenetics. We apply this to cancer in my lab, but also to other infection models in collaboration with colleagues at the Institut Pasteur. And we do this with two reasons. First, we want to use these chemical tools to better understand the molecular mechanisms that are aberrant in human diseases. Or also, and in some cases, we can use these compounds to modulate these molecular mechanisms that are aberrant and fight human diseases and develop therapeutic tools. How do we get to epigenetics? So in the year 2001, the first human genomes were completed, and we really thought that we could explain a lot of diseases based on the genome. Actually, we realized that it didn't explain where the diversity comes from and the response from the environment. If you think about it, all the cells of your body have the same DNA, the same genetic information, but a liver cell is very different from a neuron cell. We read only 2 to 5% of this DNA information that we have. And the epigenetics contribute to tell the cells which genetic information to read, like, for example, by masking certain information. And so the epigenetics defines when and where genes are expressed, and it does it without altering the DNA sequence. These are chemical modification of DNA as DNA methylation, of histone as acetylation methylation, which control the access to the DNA here represented by the blue string. In this case here, the modification makes that the chromatin is open and here is compact. So it's just a way of regulating gene expression. I'll just show you an example of DNA methylation, which is catalyzed by the DNA methyltransferases DMT. These enzymes take a methyl group from the SAM cofactor, put it on position 5 of a cytosine in CPG context. When the CPG are methylated, here represented by the red lollipop, and they're found on promoters of genes, the corresponding genes is silenced. When they're demethylated, the gene is reactivated. The advantage is that these chemical modifications are reversible, so we can change a phenotype. And this is what's really the epigenetics. From one single genome with different pattern of epigenome, we have many different phenotypes. For example, this plant, the linaria, in the north of Europe, it has these petals. In the south, these others. And the change is not genetic, but it's epigenetic. And there's in particular one gene which is differentially methylated in these two cases that make this difference. That it means also that the epigenetics transcribes the impact on the environment. And this is very important. The other advantage is that these epigenetic actors can be chemically targeted. So they are the writers. These are enzymes that catalyze the transfer of the methyl group, acetyl group. And these are, for example, DNA and histone methyltransferases, acetyltransferases. They're enzymes which erase, they remove these chemical groups. These are the demethylases, deacetylases, for example. And there's a third category of proteins, which are the readers, which recognize this chemical modification and trigger the signal. And these are chromodomains, methyl binding protein, and bromodomains. All of these three categories of protein today, there's inhibitors in the literature, and there are 9 which have been approved to treat cancer or neurological diseases. And there are many others today that are in clinical trials. So it means that we can target the epigenetic regulation in human diseases. So I will start by showing you the chemical strategy that we developed to inhibit DNA methylation. I will then show you how we use inhibitors to identify new targets as chemical probes. And then I will show you how we use these chemical tools to reprogram cells, and the aim is to develop new epigenetic drugs. I will start with the first one. So I told you that by hypermethylation of the CPGs in the promoters of genes, here represented by the red lollipop, the corresponding gene is silenced. And this is how cancer silences tumor suppressor genes. Tumor suppressor genes are the brakes of the cell. You know that cancer cells has lost their brakes and they proliferate in an uncontrolled way. Actually, the information is still there, is silenced by methylation. So the idea is to demethylate the promoters of these tumor suppressor genes and reactivate them, and so restore the brakes of the cancer cells and that recover the control. To develop new inhibitors of DNTs, we applied 4 chemical strategies. The first one, we started to pharmacomodulate known inhibitors. The second one, we did a rational approach where we coupled known inhibitors to DNA ligands to target the CPG in the promoters of tumor suppressor genes. The third one is called the B-substrate approach. And the fourth one, we set up screening assays and screened chemical natural extract library. And this gave us potent inhibitor, submicromolar inhibitors of DMT3A and micromolar inhibitors of DMT1. So I'll just detail the B-substrate approach. So it's possible because DNMTs have 2 substrates. They have the SAM cofactor here in red, who's going to give the methyl group here in green to the cytidine here in blue, which is flipped out from the double helix into the catalytic site of the DNMT. So what Ludovic did in the lab, he did analogs of the SAM, analog of the cytidine, and linked them together. He synthesized 350 compounds, and we obtained submicromolar inhibitors of DMT3A. Today, these are the most potent inhibitors against DMT3A. Here, I showed you the catalytic pocket of DMT3A. We docked in the cytidine here, who's going to be methylated, and here is the SAM with a methyl group here. You can see that our compound fits perfectly in the 2 pockets. It really is a B-substrate inhibitor. This approach can also be applied to histone methyltransferases, because histone methyltransferases, again, bind to the SAM and bind to the substrate, which in this case is arginine and lysine, who's going to be methylated. So we did the same approach, and we discovered this inhibitor, which is a micromolar inhibitor against arginine methyltransferases 4. It's a new scaffold that inhibits these arginine methyltransferases. And in collaboration with Jean Cavarelli in Strasbourg, we determined the crystal structure. And you can see here that compound 4 fits perfectly again in the 2 pockets. Here is the SAM pocket, and here is where the arginine is. Normally you can see it here from the crystal structure. So normally the arginine is here, and our compound really fits in both and make interactions which are important with amino acid to recognize the arginine and the SAM. So it's a B-substrate inhibitors. We decided then to use our inhibitors, as I told you, to try to demethylate tumor suppressor genes promoters and reactivate them in cancer cells. And here, Johan and Natasha in the lab, they treated a human colon cancer cell line. And after 3 days and 7 days of treatment, they looked at the methylation of the promoter of a tumor suppressor gene of P16. So here, the white bars is the methylation of P16. And in blue bars are inhibitors 100 nanomolar. These are very low concentration. There's no cytotoxicity. And you can see that after 7 days, our inhibitors are able to inhibit the methylation of the promoter of P16. You need to wait a certain time because these are catalytic inhibitors of DNMT. So the catalytic activity of the methyltransferase, you have to wait that the methylation that is already there is washed away during the replication cycles. The other important thing is that this demethylation is able to reactivate P16 as we wanted. So then I told you that we can use these inhibitors and make chemical probes to identify targets. And this is what Marie, Fanny, and Dany did in the lab. And the strategy is the following. So they wanted to find the target of an inhibitor, so they designed a chemical probe where the bait is the inhibitor. They attached to a cross-linking group that when the cross-link, you UV irradiate the cells that are treated with the chemical probe, it will cross-link the proteins that are bound to the probe. And then we have a handle that enable us to click to a fluorophore and a biotin to label and to purify. And then we analyze both on gel and identify the proteins by a mass spectrometry. So we were interested in this compound, which is analog of the PRMT4 inhibitors I showed you, but did not inhibit nor PRMT4 nor DNMT. We were wondering which was the target. So we linked it through a small linker to a cross-linking agent, benzophenone, and to the alkyne group. We treated the cells, we irradiated the cells, did the cross-linking with the protein targets, then we lysed the cells, we did the click reaction to attach the fluorophore and the biotin, and then analyzed by gel and by mass spectrometry. So here you see when you use an increasing concentration of the chemical probe, you can see there is bands that appear on the gel that correspond to around 50 and 100 kilodaltons. These are dependent on UV. You need UV radiation, so cross-linking to get the bands. And when you add free drug, this here, 1 and 2, you see that you delete the band, which corresponds to the fact that this is really specific of this probe. We analyze in different cell lines. So the previous one that I showed you was breast cancer cell line. Here is 3 leukemia cell line, and this is melanoma cell line. And you can see that we have some conserved proteins that are labeled with a fluorophore in the gel. By mass spectrometry, we compared all the proteins that are common, that are pulled down, purified in all the 5 cell lines, and we obtained only one, which was common to all the cell lines. And this is a chaperone protein, which uses ATP. So finally, it's not surprising that our inhibitor is acting on that. And in collaboration with Gabriel Chauzy at the Sloan Kettering in New York, we showed that this is a selective inhibitor of this chaperone protein GRP94. So finally, I want to show you how we use these chemical tools to apply to human diseases. So Arthur Schaff, who's head of the biology of host-parasite interaction laboratory at the Institut Pasteur, studies the epigenetics of Plasmodium falciparum, which is a parasite responsible of malaria. The parasite is transfected to humans via a bite of a mosquito. You see that the parasite has to survive in 2 very different environments, the mosquito, the humans. In the humans, it goes in the liver cells, in the blood cells. So it must always, to survive, adapt the phenotype. And this is controlled by the epigenetics. And Arthur was showing that it depends also on DNA methylation. So what we did, Flore in his lab and Ludovic in my lab, we screened our epigenetic chemical library against the proliferation of the parasite, Plasmodium falciparum parasite, in human red blood cells. And we obtain here the blue and the red lines, two compounds that were active against the proliferation of the parasites, comparable to the drugs that today are approved in clinics. So these are the B substrate inhibitors I talked to you about, where we did analogs of cytidine, analogs of the adenosine, and linked them together. So we have compound 20, which is active, compound 70 that are active against DMT3A, and we have also always a negative control, which is not active. So first thing that we did with Diane and Deli, we looked if there was methylation, DNA methylation in Plasmodium. And so we recovered nuclear extracts, and we saw that there is methylation here in red. This is DMSO treated nuclear extract compared to DNMT. There's methylation going on. The negative control is negative, while R2 hits are potent inhibitors of the DNA methylation of Plasmodium, while artemisinin, which is an approved drug, does not affect DNA methylation. The other advantage of these compounds is that they remain active in resistant strains. Here we have 4 multi-drug resistant convergent clinical isolates, and you can see that the compounds is active against the entire, the compounds remain active against the entire 4 isolates that are resistant to different drugs. So these are very potent to bypass resistance. We looked at the kinetics. So the parasite in the red blood cell has a cycle of 48 hours. And after 6 hours of treatment, you can see that our compounds start already killing the parasite. And they do it during the entire 48 life cycle. So these are fast-acting compounds, as artemisinin, which is today is the most potent drug. We then went in vivo. So we used a mouse model infected by Plasmodium berghei, which is the murine parasite, and that mice die by cerebral malaria death. So in green, it's our compound here. You can see that it inhibits completely the parasitemia in the mice, and it also protects from cerebral death the mice. So we have the B substrate inhibitors of DNA methylation, our new potent, fast-acting anti-malaria agent. They have a novel mechanism of action, and they remain active against resistant strain, which is really a big health problem today. So to conclude, we develop chemical tools that target the epigenetic methyltransferases. We use them to modulate a biological process, and I showed you we do that, for example, to develop new anti-malarial agents. We use them also to characterize novel mechanism, identify target, and I showed you an example of finding a new target. And I would like to summarize again the advantages of using chemical tools. They're very complementary to genetic tools. It's an advantage to use them together. In addition, we can combine chemical tools to omics approaches. They're reversible, rapid, and tunable. We can wash away the compound, and this gives really a spatial and temporal control. They allow, as I told you, to study the interactomes in cells and in vivo. And this is also very important because we know that now molecular mechanism and in any case, all the proteins involved in epigenetics are in big complexes, protein complexes. We can use them to identify new targets and new mechanisms. And I showed you also that we can apply them to develop new therapeutic agents for infection. And so I think this shows the strength of combining chemistry and biology. And I thank you. Oh, sorry. So this wouldn't be possible without my lab. So I was previously head of a lab in Toulouse. This is my attack team. Thanks to the team here at the Epigenetic Chemical Biology, we're now applying to infectious diseases and histone methyltransferases. I have to thank all the sponsors to do these research, and in particular, my collaborators, Arthur Scherf and his team at the Institut Pasteur, Odile Schiltz, who did all the proteomics, Jörg Tost, the genomic methylome that I didn't show you, and Albert Jelsch for DNMT's assays, and Antonello Mai for the diversity of the chemistry. So I'd like to thank you for your attention. So the idea, we go directly, I think, to the next speaker. That was the idea. So I will start by introducing to you Frédéric Taran, who I thank very much for having accepted. So Frédéric Taran is head of the Department of Organic Chemistry of the COA, which is the French Alternative Energy and Atomic Commission, which is located in the south of Paris. He has a PhD in chemistry of Paris University 11th, and he did a postdoctoral fellowship. He did a postdoc with Professor Barton in United States. He's permanent researcher at the COA since 1998, and he develops new regions for developing new bioorthogonal reactions to address important problems in the field of bioconjugation, labeling, imaging, and drug delivery. And so I give the screen to Frédéric to talk us about bioorthogonal chemistry with mesoionics. Thank you.
Thanks a lot. Thanks a lot, Paola, for this kind introduction. I'm going to share my screen. I hope it will work. Thanks again for this kind introduction and for the invitation. It's a real pleasure for me to present you some part of the recent results that we obtained in our lab that is located at Saclay, quite close from Paris. So what I'm going to talk to you today is about bioorthogonal reactions. So I'm quite sure that everybody knows this kind of transformations. They are really, really useful, especially if you want to link two compounds together or to cleave a molecule into two pieces in very complex media, including biological media. So in order to do that, you need very particular chemical reactions. They need to be, of course, very highly chemoselective, but also compatible with all of the functionalities that you can find in nature. So there is maybe a dozen of this kind of transformations that are very useful and efficient. And among them, maybe the most popular one are the one using azides as dipole. So of course, you know the copper catalyzed click reaction involving azide and terminal alkyne, and the copper-free version of this transformation involving cyclic alkyne. So these are very, very interesting tool, very powerful tool. And what we try to do in our lab is to try to find new chemical reactions like that, but by changing the dipole and switching from azide to other types of dipoles that are called mesoionics. These are quite old compounds that are very interesting to our point of view. So in the past, we developed several high-throughput screening methods that are really useful because you can screen hundreds and hundreds of chemical reactions per day, and even in very complex media like blood plasma or cell lysates. So you can look and screen at the bioorthogonality of a given reaction. And therefore, we prepare a library of mesoionics and combine them with different alkene and alkynes. And quite rapidly, we found some years ago now that this mesoionic called sidenones are quite interesting for click chemistry and bioorthogonal reactions. So these are quite old compounds. They have been discovered in 1935, and their reactivity has been studied by Huisgen in the 60s. So these compounds can act as dipole in the presence of alkyne, but you need to heat at quite elevated temperature. So under this harsh condition, you can form the product, which is a pyrazole product, through the first step, which is the 3+2 cycloaddition to form this bicyclic intermediate. And because of these very harsh conditions, you cannot control the regioselectivity of the first step, the 3+2 cycloaddition. And therefore, you always have a mixture of two compounds, the two regio isomers. So this is why this chemical transformation did not find so much application in organic synthesis. But what we discovered some years ago now is that if you add copper to these two reactants in the presence of this kind of ligand, batophenontroline ligand, you will have a room temperature reaction and a perfect control of the regioselectivity. So the catalytic system is about the same as the one used with azide. And of course, the product is not exactly the same. It's not a triazole as for azide. In that case, it's a pyrazole. But you have again a perfect control of the regioselectivity, and you can perform this chemical reaction at room temperature. The scope is really, really large. It's working in even with multifunctionalized reactant. It's working also in biological fluids, so it's compatible with biological media, and you can also use it for the labeling or the attaching of what you want on biomolecules. So in that case, it's here a labeling of albumin. So it's quite nice. It's a good way, I think, to synthesize pyrazole in a very mild conditions, and we can also find some nice application in the labeling of biomolecules. But the problem is the copper. We need copper, and copper, as you may know, is quite toxic. If you want to apply this kind of copper catalyzed click reaction on cells, you will have some problems because of the toxicity of the copper that might kill your cells. So this is why we were interested in developing the copper-free version of this click reaction in order to make it more bioorthogonal. So if we mix this compound, phenylsidnone, with this cyclic alkyne, BCN, it's working nicely. You have 100% of a very clean product. So it's working very, very nicely. It's working also in biological fluids. No problem with that. But the problem is the speed of this chemical reaction. In fact, it's something like 2 times slower than the equivalent reaction with azides. So we were not quite happy about that. So this is here the kinetic constant of this chemical reaction. But what is interesting with sidnone is that you can easily tune their reactivity by changing their structure. And we rapidly find that if we introduce an electron withdrawing group at this position, at the para position of this aryl moiety, and if we add an halogen at this position, and if we use a more strained alkyne, in that case, we were able to increase the speed by a huge factor. And this value of the kinetic constant is quite good, in fact, and we can now use this kind of coupling for more challenging biological applications. And this is what we try to do. So the first example that we try to do is to develop some fluorogenic probes, meaning probes that became fluorescent after the click reaction. So we developed a series of these fluorogenic probes. And today I will spoke more about this one, which is a pyridinium containing sidnone. So this compound is not so much fluorescent, but after the click, the pyrazole is highly fluorescent. As you can see here, we have a nice turn-on effect. So this is a good probe to label proteins or what you want at low concentration and in a short period of time because of the speed of this chemical reaction. But after that, we were interested in using this probe for cell imaging, and in collaboration with the group of Alain Wagner at Strasbourg, we found that this compound is able to accumulate into the mitochondria of living cells. And as you can see here, we have a huge increase of the fluorescence signal inside this mitochondria after the addition of this cyclic alkyne. So this proves that indeed we can click these two compounds together inside these living cells. But because the speed was so fast, we were wondering if we cannot use this click chemistry inside living organisms. So this is highly challenging. Of course, the chemical coupling should be highly efficient, highly bioorthogonal, but the speed is again a very important thing for the success of this kind of ligation inside living organisms. So the question was, is it fast enough for the click in vivo? So in order to address this question, we were interested in using this kind of click for a particular application, which is immuno-PET. Meaning imaging, in vivo imaging using the PET technology. So if you want to image in vivo a tumor, what you can do is to use an antibody, a specific antibody raised against your tumor. But the problem is that you cannot label it with the radio element that are currently used for PET imaging, like F18. You cannot label it at the beginning because of the short half-life of F18. And in fact, antibodies need several days to bioaccumulate at the tumor site. So you cannot label at the beginning with F18 because you will lose your signal after several days. So another strategy to label this tumor using antibody is to first inject an antibody that has been derivatized by a click element. So in our case, it's a sidnone. So you inject first your antibody with the sidnone attached to this antibody, and you wait several days, the time that you need for the antibody to bioaccumulate at the tumor site. And then several days later, you can inject your cyclic alkyne, which is labeled this one with F18. So this is a small molecule, therefore it will diffuse everywhere in the body. And if you are lucky, it will click at the tumor site where the antibody and the sidnone is. So this is what we have done in collaboration with Simon Speckler from the SHFG at Orsay Hospital. So we prepared this monoclonal antibody, which is well known. It's a therapeutical monoclonal antibody, cetuximab, and we attach some sidnone to this antibody. We injected it, and 3 days later, we injected the cyclic alkyne, which this one is labeled with F18. And this is typically the kind of image that we obtained. So this is an experiment without the antibody. So this is just the biodistribution of the cyclic alkyne. And this is with the antibody. So first injection of the antibody, and 3 days later, injection of the cyclic alkyne. So as you can see, the biodistribution is quite different. But what is really important is that at the tumor site, we have a specific signal that indeed proves that we were able to click these two compounds at the tumor site inside these living mice. So I talk quite a lot on these compounds that are called the sidenones, but sidenones belong to a very large family of compounds called the Mesoionics. And we are quite interested in exploring this chemical space by our screening methods. And today we have already identified some other Mesoionics that are quite interesting, like this one called dithiolium. So these compounds are able to undergo a nice click reaction with cyclic alkyne to form thiophene. So it's working really, really well at room temperature, and yields are always very, very good. So again, how it is working, the first step is again a 3+2 step, 3+2 cycloaddition. And after you will have a retrodisalder with a release of a COS molecule to form the thiophene. So it's working really, really well when your cyclic alkyne is not very hindered. But if you use this more strained, but also very much more hindered cyclic alkyne, you have no chemical reaction at all. So it's a kind of selectivity, let's say, for non-hindered cyclic alkyne. And we decided to develop this kind of chemical tool, a bifunctional tool, where you can recognize an azide and this dithiolium mesoionic. And if you mix this compound with these two cyclic alkyne, one which is hindered and the other one which is not, you will have only one product in a very, very good yield, which comes from the specific cyclo addition of the BCN part on the dithiolium and the DPCO part on the azide. So this is a very simple way to construct very complex molecule by attaching three partners in just mixing them, and it's a nice way to form this quite complex molecule in only one step using this double click reaction. So this is typically the kind of product that you can prepare very, very easily in in almost quantitative yields. Another mesoionic compound that I would like to talk to you today a little bit is this one called sidonimines. So this kind of mesoionic is really, really interesting, I think, because it's not only able to undergo a click chemistry, but also a release chemistry. So if you look at the chemical reaction, you will see that the cyclic alkyne will react with this dipole to form a pyrazole, like in the case of sidnone. But in the same chemical process, you will release the green part of this sidonimine moiety. So it's a click and release reaction. So the speed is not very, very high, but it's quite okay for in vitro applications. So we think that this kind of click and release chemistry can find many types of applications. And we are currently working in increasing the speed of this click and release reaction. But today I would like to talk to you about one in vitro application with this mesoionics. And this application is about the synthesis of antibody drug conjugates, ADCs. So these are very important biologics that are very, very important for the pharmaceutical industry. Several ADCs are on the market already, but there is still some need for special technologies. In particular, technologies that allows you to control perfectly the number of drugs attached to your antibody. Another need is to develop technologies that allows you to prepare antibodies with different types of drugs. So in order to address these needs, we developed this compound where you can recognize biotin, an activated ester, and the signon-imine moiety. This compound can be considered as a cleavable linker, in fact. So the technology uses an excess of antibody. So if you use an excess of antibody and only one equivalent of this activated ester, what you will get is only one antibody, well, one biotin attached to one antibody and an excess of non-derivatized antibody. Then you can add streptavidin bits to capture your antibody, and you can remove very easily by simple washing the antibody that has not been derivatized. Of course, you are not losing them because it's really expensive. You are recycling them. You keep them and you use them again in the new cycle. So once it is captured, then you can add your cyclic alkyne that has been attached to the drug you want to attach to the antibody. So the first step is a 3+2 cycloaddition, which allows you to attach the drug to the antibody. And the retrodyl solder will allow you to detach the antibody from the support. So what you have at the end is your antibody with only one drug attached to the antibody. Of course, you can use it again in a new cycle in order to attach a second drug that can be the same drug or a new drug. So this technology has been developed by a startup company that is located at Strasbourg in collaboration with Alain Wagner and two of our former PhD students. And these two young guys have developed a fully automated system that allow you to get your ADC in a very automated and robust manner. So this is the end of my talk. If you are interested in the Mesoionics, I can tell you to read this chemical review paper that we just published several weeks ago. And I would like to end this talk by, of course, thanking all of my team. I have very good PhDs and postdocs, so I would like to warmly thank all of these students. And of course, I would like to thank Davide Odizio, who helped me a lot in the training of this student. I would like to also thank Simon Speckler for all of the labeling of antibody with F18 in vivo. Alain Wagner, of course, for the collaboration that we have since a long period of time. Véronique Gouverneur for also a nice collaboration with F18, and Sébastien Papot for cell experiments, and the sponsors, and of course you for your kind attention.
Thank you very much, Frédéric. Thank you. So if you have questions, start putting in the Q&A panel. Don't hesitate. Thank you again. Very clear and very interesting and amazing, the results with the antibodies, the ADC. So now I'd like to introduce you Andrey Klymchenko, who's a CNRS researcher at the Laboratory of Bioimaging and Pathologies at the University of Strasbourg. He obtained his PhD in chemistry from the University of Kiev, and he did his postdoc in Strasbourg, then at the Catholic University of Leuven, and he joined the CNRS in 2016. He's also a fellowship of a ERC consolidator since 2015, and he leads the Nanochemistry and Bioimaging group. He is co-author of more than 200 articles and co-inventor of 9 patents. So his research interests include functionalization of fluorescent molecules and nanomaterials for biosensing, bioimaging, and diagnostic, and he will talk about bright fluorescent light-up probes for biosensing and bioimaging. Thank you very much, Andrey, for accepting the invitation, and I leave it to you, the screen.
So thank you very much, Paola, for the presentation, and thank you for inviting me to this webinar, and also MDPI, thank you for their organizing it. Today I would like to present our progress on development of new probes, and in fact, I try to see whether it switches. Yes. So in fact, in my team, nanochemistry and bioimaging, we are working on two types of chemical materials, so on molecular probes and on fluorescent nanoparticles. And so within molecular systems, we are very interested to detect and image biomembranes, RNA, and for example, membrane receptors on the surface of the cells. And with nanoparticles, because of their exceptional brightness, we are going towards detection of biomolecules in the context of diagnostics, but also for imaging in vivo, sometimes at the cellular level. So today I will be talking only about the molecular part of this work. So why we love so much fluorescence? This is a unique technique because it is highly sensitive. We can go down to detection of single molecules using microscopy setups. It is very fast. We can go up to nanoseconds and picoseconds because the process of fluorescence is very fast. Then it is non-invasive because you don't touch the sample, you just shine light and you collect light. And it is specific because we, in principle, can address molecules by so-called fluorescent probes. And therefore, with this technique, we can go from single molecule and cells to tissues and up to animals. And the idea of a fluorescent probe, and especially what we are working on, is to have a system, a molecule which responds to the target and to respond specifically. And here you have two types of response. One can be fluorogenic. So Frédéric Taran was already mentioning fluorogenic system, where after binding to the target, or for instance, click reaction, the molecule lights up. The other are chromogenic systems, where after binding to the target, they change their color. And so I will present both systems, but our approach is not based on the chemical reaction, but on the recognition of the environment. And what happens when you put a molecule which is sensitive to environment, for example, it can have a different color or different fluorescence intensity because of its interaction with the water molecules. Once it binds to the target, the environment can change. For example, receptor or membrane, lipid membrane, the environment change, for example, from water to apolar viscous oil-like. And this would lead to changes in the color, intensity, etc. That can be used for detecting practically any sort of interaction and for bioimaging. And this I will show you. And what kind of environment-sensitive molecules exist? I give you some examples, major, that are used in biosensing and bioimaging. So one is based on so-called excited state charge transfer. These molecules are dipolar and they are sensitive to polarity. Then you have twisted intramolecular charge transfer system where a molecule rotates and it actually increases intensity on increase in the viscosity of environment. And then you have aggregation cost quenching system where you have a dimerization or multimerization of molecules. And so when they're aggregated, they're non-fluorescent. And when they disaggregate, they become fluorescent. And this can be all used in biosensing. So, and let's go to chromogenic system based on solvatochromism. So solvatochromism, what's that? So dipolar molecule in the excited state, actually, it increases its charge separation. And because of its interaction with high strong dipoles, for example, of water, the energy becomes lower. And this decrease in the energy is translated into change in the color. And so in more polar environments, it becomes rather red emissive. In a polar oil-like, it is blue. And this can be used for detection of biomolecules or especially biological membranes. Why? Because it is composed of lipids which are highly apolar. So the example is a red molecule. So it's sensitive to polarity on change in the solvent, it shifts to the red on polarity increase. Now, when you add it from water to lipid membranes, when it binds to lipid membranes, in water it emits in red and intensity is low. And when it binds to lipids, it changes its color, so energy goes up, intensity increases, and there is a blue shift. And this turn on and the blue shift is actually used for sensing. I will show you how. But first, when we want to image something in the cell, we need to, in a living cell, we need to target it specifically. So, example, if you take an red which is non-functionalized, you add it to the cell, you see all this non-specific binding, you see fluorescence from everywhere, and you see these dots, these are lipid droplets, and all sorts of membranes of different organelles are fluorescent. So to have more specificity, for example, to cell surface, to cell membrane, plasma membrane, we added a special anchor group, which is a ZVTRN with a long chain, and you can see now we can target specifically plasma membrane. So that's all right. And now we can exploit solvatochromism. Solvatochromism can sense, for example, lipid organization on this membrane. You have on the membrane can be classified to two types of phases. One is loose phase, which is liquid disordered phase composed of unsaturated lipids. And another is liquid ordered phase, is composed of saturated lipids and cholesterol. So this is highly dense phase, and therefore there is no water, there's not much water in it, so it is highly apolar. And this phase is polar. So normally solvatochromic dye could distinguish it by its color because polarity is different. And it is the case. For example, when you do imaging of with this probe, when you add the probe to disordered phase, you have one emission color, and to ordered phase another emission color. But careful, this color is actually pseudo color, which reflects actually the ratio of the two colors. So when the band shifts, the ratio of the red and green part of the spectrum changes, and so this is converted into the pseudo color. I hope you see all the screen because I have here the square, but I think it does not. I hope it does not hide.
No, you don't. It's my same problem. You see, we see your screen complete.
Yeah. Okay. Then it's fine. Perfect. Thank you. Yeah. So, and then when you have, for example, mixture of ordered disordered phases, which are so-called models of lipid rafts, you could nicely see separated phases on the model membrane. This is a model giant physical membrane. And then when you go to cells, you can do the same by imaging ratio on the cell surface, but here you do not see any phase separation. You see actually a continuous phase. But when you extract cholesterol, for example, the phase becomes more and more disordered. So you remove cholesterol, the membrane becomes more disordered and the color changes. Okay, this was early work back in 2010. And then we asked a question, why we cannot see these rafts? And we thought it would be interesting actually to develop a technique where we can detect with high resolution the membrane surface. And for that, you need to have a molecule that blinks. And to achieve blinking, so non-red dyes, they are not really capable to blink because they just bleach when you apply strong light. So we need to generate an object which can blink, and that could be done by reversible binding. And this we have done by using an analog of our initial molecule, which has a short chain, hydrophilic chain. So it will have a low affinity to membrane, so we expected it to bind reversibly. And as you can see in the video, these points are single molecules. And so reversible binding is observed as just appearance of those spikes and disappearance. So it's a binding and unbinding events. And in fact, in case of our original molecule, which has high affinity and binds irreversibly, you don't have any blinking process. And so thanks to this blinking, we can do a super resolution imaging. It's so-called single molecule localization microscopy, where we, thanks to the reversible binding, we can reconstruct the super resolution image of the cell. This would not be possible with a classical probe that binds irreversibly. And then having high resolution on the cell surface, we also tried to see the color of the probe, and we could see some objects on the surface, the nanometric size on the surface of the cells, which are actually perturbation of the membrane, like invaginations and fiber-like structures, which are characterized by higher polarity and actually lower lipid order. And so what we concluded that when membrane bends, it creates sort of disorder, but at the nanometric scale, and we can detect it. Okay, but that was for the cell surface. So how about inside the cell? Can we target specifically with those environment-sensitive probe intracellular environment? So this, I showed you example of the cell surface, but we can also target lysosome by adding a protonatable group. We can target lipid droplets by adding hydrophobic group. Then by adding a charged group, we could go to mitochondria, as Frédéric was showing for another derivative, which should be cationic. Then we added some other groups, which are reactive groups, to go to ER. And also by lipophilic group, we can also target Golgi. And this design allows us to target membrane, you could already see, that it targets membrane. And then we do colocalization with a reference marker of the given organelle. And you can see for ER, commercial marker and our molecules bind the same way. So they target ER. Mitochondrial one colocalize also with the mitotracker. Then lysosome-based molecule colocalizes well with lysotracker. And then the same for Golgi, very good. And lipid droplets also colocalize with corresponding tracker. So all this chemical targeting loop allows you to target NIRAD to the place you want. But now, as this NIRAD is sensitive to polarity, you can extract the information about the color. So by recording the two emission channel and dividing them, we could extract the pseudocolor, which will reflect polarity of environment. For example, if you take lipid vesicles and do image, this is ordered phase will appear in one color and disordered phase in another. And like this, when we look at this color, we can interpret. So this is rather corresponding to ordered phase, and this one to low polar phase, etc. And this one to relatively polar disordered phase. So what we actually found out that different organelles have very different environment polarity and therefore different lipid order. And in fact, the most ordered phases was on the cell surface, while the most disordered phases was in endoplasmic reticulum. And in this respect, having this sensitive tool at different organelles, we ask ourselves, can we see some processes, for example, like lipid peroxidation in the presence of reactive oxygen species? So lipids can undergo oxidation, and this should change polarity. And in fact, what we found out that different organelles react differently to the action of hydrogen peroxide. And so some, for example, like lipid droplets and lysosomes were very sensitive, and mitochondria, while others were less sensitive. So it depends on lipid composition. And of course, also on resistance of cell to the oxidative stress. And so then I would like to show you an example of the probe operating by the mechanism called molecular rotors, where the molecule rotates. And in this case, the examples of molecular rotors, these are also usually push-pull molecules, but they have free points of rotation. And this, due to this rotation in solution, they are not emissive, but when you increase viscosity, the fluorescence goes up. And this was actually used, for example, for imaging organelles, but in this case, viscosity in different organelles of the cells. And what we were interested to develop fluorogenic system to detect a specific lipid on the cell surface. And which lipid we selected, it was phosphatidylserine. And phosphatidylserine, it is interesting because in normal cells, it is mainly present on the inner leaflet of the membrane, while on apoptosis, it is getting exposed on the outer leaflet. And if we design a molecule that specifically detects phosphatidylserine, we can detect apoptosis. And for that purpose, we actually took a receptor which was known in literature based on the picolylamine unit, and this can complex zinc. And this receptor can bind specifically to phosphatidylserine. We couple it to this molecular rotor, and this molecule is quite interesting to show you the synthetic route. So there are many steps. First, we have to build the body pi unit, and this was the acid body pi, which was then coupled to the linker, PEG linker through, and we used click chemistry, of course, because when you build really a big molecule, then we build two parts and then we connect them by click chemistry. The second part was the receptor with typical ethylamine and corresponding alkene. And then by copper catalyzed click, we got the molecule and the zinc complex. So when you add this to the cells, which are treated with the agent to generate apoptosis, what we could see, in fact, we could see that this molecule can bind to certain cells. To understand whether they bind to apoptotic cells or not, we used another marker, which is called annexin. It binds specifically to apoptotic cells. And we could see a colocalization of our probe, which is in green, with annexin, which is in red. And other cells which are alive, which are not yet apoptotic, they did not show any fluorescence. So we could detect apoptotic cells, but also more we could see late apoptosis. When cells start dying, the probe can internalize, and this was not the case of annexin. So we could distinguish even early and late apoptosis. So we went further and we say, okay, now we want to develop something brighter. And to get brighter, you need to bring several dyes together. And to have a fluorogenic response, we considered a system which is dimeric and which turns on when it binds to the target by opening. And here we target a receptor on the membrane. And so to build this dimeric system, we combine it through a lysine group, which through the linkers, and this which selected squalene molecule, which is known to have very high extinction coefficient. So now we put two squalene molecules, which give you a huge extinction coefficient with high quantum yield. So we have a system which is almost as bright as some nanoparticles like quantum dots. Okay, and then this molecule indeed works by closing an opening, by changing the environment. In water, you have a characteristic band of the dimer, so-called H aggregate, a blue shifted band. And when you go to methanol, it opens up and you get a normal spectrum. And this can be seen actually in fluorescence. When you increase the amount of methanol, the fluorescence increases. For the monomer, the fluorescence changes very little bit. So we have a system that opens up and closes when we change the environment. And now to exploit that, what we did, we coupled this, our dimer to a ligand. It's a cyclic peptide, which targets G protein couple receptor. So this was done in collaboration with Dominique Bonnet in Strasbourg. And what we did, we targeted G protein couple receptors of oxytocin on the cell surface. And so when you add this molecule, you could see nicely the fluorescence on the membrane. So we target the membrane. And when we used a similar probe, not a similar probe based on rhodamine, which is always on, you could see a strong background. So the probe will always on, so you see fluorescence everywhere on the surface of the cell. So it targets the cell surface, you can see fluorescence, but you also see it outside. So this shows the importance of fluorogenicity for imaging. And another important thing, even more important, when you go in vivo. And because in vivo, when you inject in the mice, you will see fluorescence everywhere, but we want to see fluorescence only in the target. And for that, we did a dimer, which was near-infrared dimer. So this is another work in collaboration with our colleague, Dominique, and based on cyanide 5.5 in this case. So the molecule works in a similar way, but it operates in near-infrared, which is important for in vivo imaging because tissue is more transparent. And so when we injected in lactating mice, which present oxytocin receptor and the mammary glands, you can actually identify them on the image. And in fact, if you add free ligand to replace it, you see there is no more fluorescence. And so that was, and also non-lactating mice shows poor fluorescence in this mammary glands region. So we could directly see receptor natively expressed in mice. Okay, and we can detect not only lipids and proteins, but also nucleic acids with this approach, with the environment sensitivity, including dimers. And in fact, what was done previously by Jeffrey Group in the US, they showed that by taking a molecule, which is like kind of molecular rotor, which is non-emissive in water, you can detect an aptamer molecule, a nucleic acid as RNA, which folded in such a specific way that it has a binding site for this dye. When it binds, the molecule stops rotating and it shines light, and it's a GFP-like fluorophore. And what we took in literature, look in literature, and we found that there is aptamer for rhodamine, but rhodamine is always fluorescent. And we wanted to have a fluorogenic system that turns on binding to aptamer. So therefore, we made this dimer, which behave exactly the same as I show you for another dimer. It also shows you a specific band, blue band, and poor emission in solution because it dimerizes. And then we run, in collaboration with Michael Rickelik in Strasbourg, we run a selection. There was a mutation of this RNA aptamer and so-called molecular evolution. And millions of molecules were tested. And then we found out that during selection process, the nature generated a dimeric aptamer. And this dimeric aptamer interacts with this dimeric structure and opens it up and lights it up. And in fact, this works in cells. When you look at in the cell expressing this RNA, and we add our molecule called Gemini, we can see this fluorescence in red is actually fluorescence of RNA. And if you express only GFP, you see only green fluorescent protein, you see no fluorescence. So, and in fact, because we use rhodamine molecule, this system is very photostable. So when you shine light for a long time, you have no loss of fluorescence or weak loss of fluorescence, while other molecules which are currently used with this technique, they are not very photostable. Okay, and finally, I want to show we can go beyond even the dimer and go to more molecules. It becomes more complex. If you want to get a very bright signal beyond the single molecule, why not to make a dendrimer? And that turns out to be not so easy, but that we managed to make. So because the idea is not to have a dimer, but many units that can turn on by opening. And for that, we did different dendrimer, and we found out that dendrimer, which has long linkers, that one works very well. And in fact, this one has a very strong turn on when you change from water, where it is self-quenched because it is collapsed, to a polar solvent like dioxane, where it opens up. So you have like 400-fold increase. And this molecule has a very high extinction coefficient, of course, because it's 8 units. And then, of course, the synthesis is of course not obvious, but we need to synthesize first a unit, reactive unit that will react with our dendrimer containing amine. So scoring that was coupled then after to amino dendrimer. Okay, these are BOC. We remove BOC, we coupled, and we got this molecule. And then we had to graft also RGD for specific targeting. That was done again with click chemistry. Very important to use click chemistry at the end when the molecule is complex. And then we added to the cells, and you can actually see for the cells presenting integrin receptor, RGD targets integrin on the surface of the cell. You can see nice internalization of the probe and fluorescence. In case of free RGD ligands, it replaces, so it cannot accumulate. And if you use the same probe dendrimer without the specific ligand, there is no targeting, and the monomer actually is everywhere because it's always on. Okay, so I presented you a little bit of overview on what we are working on the molecular level, and you could see that we can generate response to the target or to the environment through different mechanisms. Solvatochromic dyes we use a lot now. Red, we use other dipolar molecules, but I didn't have time to show. We can target different organelles and study specifically polarity in the cell. Then molecular rotors, which rotates with intramolecular rotation, can be used to turn on when the target is more viscous. So this was used to detect, for example, apoptosis. And then fluorogenic systems based on dimers and multimers. Those are very bright systems that can detect, for example, receptors, membrane receptors, or intracellular nucleic acids. And of course, here we go to multifluorophoric systems, and that's actually explained why we also work on nanoparticles, where we put a lot of molecules together in one particle, but this is another story. Okay, and I would like to thank to my team and of course to the people who worked with me for long years, Mayol, Andras, and Philippe. There were lots of PhD students and postdocs who contributed to this work, and biophotonics unit with whom we collaborate on some microscopy applications, and of course collaborators, external collaborators who worked on more specific applications like on receptors, on super resolution imaging, on the RNA detection, etc. And thank you for your attention. Of course, the financial support, shouldn't forget ERC grant, of course. And so now, thanks.
Thank you very much. Thank you very much for this clear presentation, an amazing amount of tools, fluorescent probes. So first, maybe for Frédéric, I had a question on the thiophene and the ones that release the thiophene probes. Did you apply them to living cells? Are you going to apply them in biological setup? So if I understand well, they release thiophenes probes and then COS, right?
Yes, yes, right. Well, at the moment we do not use this click chemistry in living cells or in for biological application because the speed is not. So the kinetic constant is 0.1, and so it means that you need several hours and even days if you are dealing with very low concentrations. So so I think it's quite nice for in vitro applications and for the constructions of quite complex molecules, but their use in cells or in vivo, I think we need to improve the speed.
Okay, yeah, because yeah, it was useful. You can use two different probes. The fact that it is the one with the less steric hindred, right? Cyclooctanes. Yeah. Okay. Thank you. You have a question. So Frédéric, great talk from Mark Brunstrup. How complex is the synthesis of the Mesoionic?
Oh, it depends of the Mesoionics. The sequence is really simple. You start from an amino acid, and in one step you can get the signal. There is one step which is the nitrosylation of the amine. So this is not really cool. But we develop a one-pot process which allows you to not be obliged to isolate the nitrosoamine, which is really toxic. So you can form the signal in one step, no problem really. The deuterium are more tricky to prepare. So this is also a reason why we are not so much working on this Mesoionic now. So it's, if I remember well, 4 or 5 steps, and the yields are not so easy. Signonimines, that's quite okay. Again, we need 3 or 4 steps depending on the signonimine. And now we are working on the improvement of the optimization of this synthesis. So signon easy, deuterium both well not so good, and signonimine quite easy now. And there is plenty of other Mesoionics. Some of them we have already published. And I would say that usually there is at least from 1 step to 3, 4 steps.
So are they explosive like azide?
Well, I don't think so. We never had any problems. We worked some years ago on a Mesoionic that so it's a 5-member heterocycle, and this 5-member heterocycle we had 4 nitrogen. So we were very scared about about the explosivity of this compound, but in fact, no problem. These compounds are quite stable. So
important argument with respect to azides, because azides really can be dangerous. Of course, for small molecules, for big molecules, it's no more important. But one question is. So when you're talking, I have questions also to Paola, but as I started with Frédéric, I have a question about in vivo application. So you said you need to have a very fast reaction, but it's not only that, because when you inject in the blood circulation, you should be sure that your molecule A and molecule B will be bound to the same thing, because they will always bind somewhere. They will not be in solution. They will bind to BSA, they will bind to lipoproteins or to the cell. So, did you study this in complex biological media before injecting? So how would you bring them together in complex media?
Indeed, this is of course a very important question. Of course, the speed is not the only point. Well, in fact, what is really difficult to manage is the biodistributions of the two partners. So, yes, we are trying to improve this by using targeting strategies. In particular, for cancer, what you can do is to attach something that allows you to concentrate the two partners at the tumor site, let's say. So of course, it's not perfect. There is no targeting strategies that are really, really perfect. In fact, it's quite far from perfect, but at least you can concentrate a little bit your partners at the tumor site. And so this is the thing that we are currently doing now.
Even if you are in a tumor site, you have to be sure that within the tumor, they are located within similar organelle or within lipid or within a cytosol, so that they can meet. And if one is in mitochondria and another in Golgi or something like this, they will never meet.
Exactly. So it's a kind of black box at the moment. We have no perfect strategy to deal with that. It's really, really complicated.
Okay.
And Andrey, concerning this question, for you, when you showed your probes, your solvent chromatic probes, you were doing it on fixed cells, right? You don't do on live cells.
On live cells. On live cells, because solvatochromic probes here, we want to see the native organization of lipid membranes. And so, for instance, in mitochondria or in Golgi or in the lysosomes, if you start fixing, you will disturb the native organization of lipid membranes. And in fact, you start permeabilizing the cell. So lipids are no more the same. And in fact, you can then follow the process of, for example, the cell death or the stress process, even mechanosensing you can do. And this is done on the live cell. Because lipids, they don't like fixation. They don't like any sort of disturbance. In fact, if you look at their cell death markers, the cell is considered to be dead when the membrane becomes permeable. And in fact, this is typical lipids, they react immediately. And lipid organization. Yeah.
But I was surprised because they're quite huge molecules, so they enter without any problem then in live cells.
Oh, everything is relative. You know, we work on particles which are 10, 20, 30 nanometer size. So this is still 1, 2 nanometer. And yes, molecules, even this dimer, that was very big surprise. The dimer of rhodamine was able to enter the cell, but it is entering slowly. It enters on there within 5 minutes, 10 minutes. But if you take NANRAD, it enters on millisecond time scale. It's just a diffusion. It goes like a knife into butter. There is no any barrier. And it's very fast. For markers of organelles, yes, it takes minutes to accumulate in a given organelle. Yeah, so that's sure. It's it takes time, and especially when you want to have a specific distribution. First, it goes everywhere, then it accumulates. For example, mitochondria is driven by membrane potential, And so it needs to accumulate slowly. It's the same for lysosome, it's driven by protonation. So it will protonate, it will accumulate, and after a certain time, you see distribution towards one organelle. Yeah.
Thank you.
And I have a question to Paola about, you used SAM, this molecule, which is intermediate, which is substrate for methylation. And in fact, I was wondering whether SAM, can it be used as a, because this is a kind of metabolite, right? And can it be used in diagnostics applications and to see whether, because methylations are frequently related to some anomalous processes, pathologies, can it be a marker of diseases?
So actually what they measure is the SAH, which is the product of the reaction of methylation. And some diseases, as they have too much, for example, SAH, it happens. So yes, it's something that is followed. But the SAM is quite unstable molecule, and so they measure rather the SAH. The product of the reaction.
SAH is a product of methylation?
Yes.
Okay.
So it's the adenosine.
Okay. Yes. Okay. I see. And another thing. Okay, this is very how to say popular question, I think. You were mentioning the pathology like this, with this pathogen depends on the epigenetics. This pathological process was asymptomatic and not symptomatic. So the obvious question is whether I guess there is a strong research also on the effect of epigenetic expression in humans and its response to COVID disease. Is there? I guess there is a lot of research on that, right?
So there's the research for infectious disease and epigenetics is starting. It's something quite new. And we were lucky because the compounds that are active in the malaria and the parasite are inactive in humans. So we had we're lucky because the IC50 is really completely different.
But so for response to virus, it should be, because humans respond so differently to COVID. And I guess it's also related to...
For epigenetics, the sensitivity, yeah, for sure, because it regulates the expression. So definitely. So I'm sure that there are studies going on for sure. And there are studies going on trying to understand the predisposition of patients and on their how their epigenome will dictate their answer. So there's studies going on there for sure. Okay. There's a question for you, Andres, from Gracidavas Lukinavicius. So Andrew, next presentation and set of the probes. My question is about fluorogenicity. Do you need it for the membrane probes? High selectivity and affinity should lead to high local concentration, which should provide good contrast in microscopy. Maybe you need to improve targeting ligands in addition. Can you comment about this? Thanks.
Yeah. Thank you. Thank you, Gracidas, for the question. I know you, yes, he's also working on fluorogenic system. So the question is, he knows the answer. It's very important to have fluorogenic system. But on the other hand, solution can be to have something with high affinity. And so when you have a molecule which targets very specifically with high affinity to its target, fluorogenicity is less important because you can go down with concentration, and so you can specifically target and then wash even if you want. But in no wash conditions for the targets which affinity is less high, then it is absolutely important to have fluorogenicity. And also in no wash conditions, you apply a certain concentration of dye. Starting above 10-20 nanomolar concentration, you will have a background. So therefore, fluorogenicity is important. But if for membranes, in fact, the question is whether the membrane probe needs fluorogenicity. Yes, absolutely, because in fact, in microscopy, you can see something without fluorogenicity, but very difficult. But in solution, when you try to probe for example, cells in suspension using spectroscopy, without fluorogenic response to membranes, impossible to see because you will see only the bulk solution. So, so this means in, for example, future high-throughput screening assays, if you want to detect target in solution, you need fluorogenicity. In imaging, it's maybe a little bit less important, but it will always help to have turn-on system, because you will see only the target and you will not see others. And in fact, fluorogenicity is absolutely important for super resolution imaging that I presented. It is so-called paid system, where the molecule is non-fluorescent. When it binds, it turns on, and you have this switching behavior. This is where you also need fluorogenicity, where otherwise it won't work, this paint blinking process. Okay, thank you.
I had a question again for Frédéric. So I liked your click and release and how you applied it to the IDC, which is quite impressive. So you have really a turnover, if I understood, because you can first recover the antibody, and you recover also the streptavidin that enables you to purify, and so you can start the cycle. Is that the idea? You always read the cycle.
Yes, exactly. So so first you capture the antibody, but we use an excess of antibody. So you capture only a small part of the antibody with only one biotin, and the other three antibody you will recover them in a flask, and you use them in a new cycle. So at the end, you have all of your antibody with only one biotin. Then you inject this antibody into a column where you have your streptavidin bits. So you capture this antibody, and in this column you add your cyclic alkyne with the drug, and so you have the click and release, and so you can recover your antibody with only one drug, and then you can use them again in a new cycle, injection into a new another column with the cyclic alkyne with a new drug if you want. So it's a way to control what is called the DAR, the drug antibody ratio, and you can control the number of drugs and the nature of the drug. And so this has been developed in a Strasbourg lab, in the startup, as I mentioned in my talk, and they develop a fully automated system. So now we just have to push a button to get the ADC.
And they tried different drugs, or they always?
Yes. In the paper they published, I don't know, maybe dozens of ADCs with different drugs, different proteins. It's not, of course, working only on antibody. It's working on any kind of
protein.
Of protein and any kind of drugs. They also use this technology to attach not only drugs but also nucleic acids.
Yeah, it's very versatile, and it was really, it's impressive, this click and release. Very neat. Thank you. Andrei, you had a question, I think.
Actually, yes, I had a question to Frédéric about in vivo experiments. It's always tricky, all these in vivo studies, and you did a 3 days incubation. Why you waited so long, and whether within 3 days are the antibodies still alive? I mean, there can be some biodegradation during this period.
Well, in fact, antibodies, they have a very particular pharmacokinetic in vivo. So, in fact, they stay for something like 20 days in the body before being destroyed by the catabolism. So they are quite stable, in fact, and they circulate in the blood, in the body, for several days. So this is why you need a long period of time to have enough antibody at the tumor site. So if you inject a given antibody raised against a receptor that is expressed in a particular tumor, many time you will have only from, well, let's say around 10% of the injected dose that is at the tumor site. And to reach this number, you will need 1, 2, 3 days. And then you have reached the plateau of the bioaccumulation at the tumor site. And the 90% other are still and so on. So it's a very slow pharmacokinetic.
Wow, well, very impressive. I didn't realize. I guess there are studies on the elimination time of antibodies from blood and also on biodegradation time. I guess it's not so difficult to do for after injecting antibody. It takes several days. It can survive. And yeah. Okay, okay. It's very, very surprising, but okay. Yeah, we usually 3 days. We don't do 3 days experiment on this.
No, for sure. Well, with a small molecule, it's really fast. In a few minutes, the compound is everywhere, and but with well, antibodies, it's 150,000. The mass is 150,000. It's a very huge, so the pharmacokinetic is completely different when you compare it to regular drugs.
And one question to Paola is about this molecule inhibitors of their methylation. The thing is that I mean, you are inhibiting practically any methylation process that will take place in the cell, right? Or it will inhibit only certain process that you, which is dependent on the sequence of nucleic acid. Because, for example, with CRISPR, you can control where you do things. But how with inhibitors, how selective they would be? And I mean, the question is how toxic will be then, teratoxic commonly, how it is called there, teratogenicity of this.
Teratogenicity. So actually, it's important to note that they are inhibitors of the enzyme when it's methylating. So what we observe in cancer cell, there's a specific hypermethylation. There's an hyperactivity of the DNMTs. So our inhibitors really act when there's this a lot of methylation going on. So they won't. It was surprising. We saw it by looking at the methylome, global methylome. We found really specifically acting on the promoters of the tumor suppressor genes. And depending on the cell lines, you have different profiles because it's not the same where the activity of the enzyme is. So it's really dependent on this hyperactivity of methylation, which is specific. So they're not sequence specific. It's something interesting. And actually where we're moving rather to try to target the partners that bring the DNA methylation to these specific sequences. So we're trying to identify the partners and who is targeting them. But for the moment, they're surprisingly specific because you need this hyperactivity of DNA methylation.
Okay, so methylation is not happening intensively in the cell, it happens during pathological process or...
Yes, no, there is also normal cells, but it's kept at a certain level. Here we see really very specific, in cancer cells you have a loss of DNA methylation and a very specific activity going on there. So there are patients that are since 20 years on one drug, which is called 5-azacitidine, for example. They don't have side effects.
But you said it's reversible, so you can demethylate?
Oh yeah, it's reversible. You can wash away and it comes back.
But did you check that if you block methylation, maybe you are kind of demethylating also? Because everything is equilibrium. I mean, in the cell may react to it by doing demethylate. No?
Well, actually, our effect is transient. Then it goes back up. So there's something going on. So for us, there's really a window where we should treat with DNA inhibitors and then combine with other drugs to kill the cells there. And that would be the idea. So there's 2 questions. Thank you, Andrey, for the question. Frédéric, thanks for the nice talk. It's Fei-Liu. I would like to ask which concentration you did to the antibody click and release reaction. I mean, which ratio of antibody and reaction counterpart, because antibody is a huge protein.
Yes. Alors, so concerning the ratio, so the first step we use an excess of antibody, as I was saying an excess of antibody and a tiny amount of the synonymine that should be linked to the antibody with the biotin part. And then after the capture, we use an excess of the cyclic alkyne and the drug. So, but this is not really, well, this is okay because we are recycling everything. So we are losing nothing. I'm not sure if I'm clear, but so the first step is the derivatization of the antibody with the biotin. So we use an excess of antibody in that case, a huge excess in fact, to form only 3 antibody and one antibody with 1 biotin. So this is with an excess of antibody compared to the activated ester that we are linking to the antibody. And once it is captured, we use an excess of the cyclic alkyne, which was attached to the drug, in order to be sure to have 100% of the cleavage of the immunocidin linker and the attachment of the drug. And so we use sometimes an excess of antibody, sometimes an excess of cyclic alkyne. But what I would like to say is that it's not really important because we are recycling everything. So we are losing nothing.
Well,
good for the company to economize. Huh?
Exactly, it's really, really expensive. Everything is expensive. The antibody, the
drugs.
The cyclic alkyne, the drug, everything is really, really expensive. So we need to lose nothing.
So, well, thank you very much. I think it was a really nice discussion, beautiful presentation. So thank you very much to having participated and accepted. And thank you very much also to the participants. So please, I saw that there were many French participants, so please don't hesitate to contribute if you want to the issue. And thank you again also to the Molecules for the organizing the webinar. And I don't know, maybe Cedric, you want to add something? I saw you pop out.
Yes, thanks. I would like to thank the chair and the speakers for these wonderful talks and for running this webinar so smoothly. Thanks to all attendees for participating in today's webinar. Thank you very much for your time. Have a nice day or even wherever you are around the world. And also, of course, a good weekend. Take care and please stay safe.
Yeah, thank you very much.
Bye.
Thank you very much.
Thank you.
Bye-bye.